Hello students, we were on the chapter Human Health and Diseases. Right students? In the previous lecture, we have already talked about the various kinds of immunity, different immune system, and even we have talked about the innate and acquired immune system. Hope you have completed all that topics. And we have already talked about the different immune response, that is a primary immune response, the secondary immune response, even we have completed the cell mediated immunity and the humoral immunity, right? And hope you all are cleared with what are B cells, what are T cells, what are the different type of T cells, what are antibodies, right? Like in previous class, we were discussing the structure of antibodies and how the phagocytosis occur, right? So in this class, we will be talking about one important topic, that is vaccination, right? Vaccination. Students, it's a very important topic, vaccination. First of all, before going in detail, I would like to tell you who was the father of immunology. Father of immunology. Who was the father of immunology? Very important. This you have to write in those notes, the father's notes. So hope you have maintained that notes. The father of immunology is Edward Jenner. Edward Jenner is considered as father of immunology. Now, why is he considered as a father of immunology? Just because of one term, that is a vaccination. Vaccination was started by the Edward Jenner, and he has created the vaccine for smallpox. So what he has done, basically he has seen that whenever we incorporate the inactive form of the smallpox vaccine into the a normal person, so in the subsequent ages or subsequent time, what will happen, the person become immune to this one particular disease, that is a smallpox, and later on when the infection will occur, the person will not show any kind of symptoms, right? On this basis, he was considered as a father of immunology. Who was the father of immunology? Edward Jenner, right students? And he worked on the smallpox, right? He has created the vaccine for smallpox. Now, the question arises, what do you mean by vaccination? Vaccination or creation of vaccine. What are vaccines? Vaccines are inactive form of pathogens, right students? We have already discussed that what do you mean by inactive form, what do you mean by active form. We have already discussed those things. So vaccines are inactive form of pathogen. Then why we use this inactive form, right? This is very important, inactive form, right? Now what do you mean by inactive form? Inactive form means the pathogen or that virus is in inactive state. If we incorporate these vaccine or these vaccine in the active form, right? Now what will happen? There are chances, there will be chances that the person will get the such disease which are associated with that pathogen, right? The main focus of this incorporation of inactive form is to create a primary response and the memory cells, right? So the vaccines are the inactive form of pathogen which are which are injected into human or into humans. Not only for humans, vaccines are available for different vertebrates as well. So this is the basic definition of vaccines, right? There are so many types of vaccines available. There are certain vaccines like first generation vaccine, second generation vaccine and third generation vaccines. So we will be doing that. First of all, the vaccines are the inactive form of pathogens which are injected into the humans, right? And these inactive form, this inactive form of pathogen can be in the form of attenuated, like they are heat killed, or sometime what we do, we use the surface antigen, right? Suppose this is a antigen, we use some of the surface antigen. This is a pathogen and we use a surface antigen, and that surface antigen this is considered as a vaccine and that is incorporated into the body, right students? So this was vaccines. Now we will be taking what is the principle behind vaccination. Very important, principle behind vaccination. As we have already discussed, the principle behind vaccination is the creation of primary immune response and the memory cells. Right, students? So, the principle of vaccination. Principle of vaccination. First of all, why we insert the inactive pathogen into the body? When we insert the inactive pathogen into the body, then what will happen? There is creation of primary immune response and the creation of memory cells. And these memory cells are very important in terms whenever the second infection will occur, the time of recovery will be less as compared to the primary infection, right? So, basic principle behind vaccination is that whenever, we can write when we inject inactive, this inactive form can be heat killed form or a very small antigen inactive form or surface antigen. Another important word, that is a surface antigen, right? The surface antigen is inserted into the body when we inject the inactive form or the surface antigen of pathogen there will be primary response, right? The primary response will occur because primary response in this case is not necessary because this is also an inactive form, right? As this is an inactive form, this will not cause any disease, so we are not concerned here with the primary response. With the primary response, and the most important, students, and formation of memory cells. Here this memory cells are very important. And the formation of these memory cells. So, after the memory cells formation, what will happen when the person will encounter the same antigen for the next time, right? Students, come at this side. So, this will be easily visible to you. So, when person will encounter the pathogen, for second time, or you can say the first time. This is a basic first time because the patient or the person has already encountered with the antigen, but that particular encounterance is with the inactive form. Now, when it encountered the pathogen for second time, right? In this is second time, suppose the pathogen is active. Pathogen is active, right? There will be formation of secondary immune response. For second time, there will be secondary immune response. Secondary immune response, right? In this case, what is important? The secondary immune response. At the end, what we need is a secondary immune response, right? Very important, secondary immune response. And why this is important? Because this immune response is comparatively faster as compared to the primary immune response. In the primary immune response, what happens is the patient experience some disease, or some patient may experience some disease whenever the pathogen is active, right? In this case, when the second time the pathogen is active, now what will happen? The antibodies are already there. The main thing is that the memory cells are there. So when it will encounter the pathogen for the second time, now what will happen? The secondary immune response will occur, which is much faster than the primary immune response, and there will be eradication of the pathogen from the body and in a very fast manner, right? So this was the basic principle of vaccination, right, students? Students, come at this side. Now we will be talking about different kind of vaccines. I've already told you there are different kinds of vaccine. Those are first generation vaccines, second generation vaccine, third generation vaccines, right? In some books only 2 generations are there, but actually there are 3 generation vaccines. Types of vaccines, right, students? Types of vaccine, there are 3 types. The first, we will be talking about the first generation vaccine. First generation vaccines. As the name indicates, first generation vaccines. In this, these are those vaccines. These are those vaccines which are created by inactive form of pathogen, or what we do, we inactivate those pathogens, right? Vaccines which are formed by inactivation of pathogen or by using surface antigens, or by using surface antigens of pathogens. Right, students? What was the first generation? What are the first generation vaccines? These are the vaccines which are formed by, in this, what happened, this process occurs, that is an inactivation of pathogen and by using the surface antigen of pathogen. As the name indicates, the inactivation here means we have used the whole pathogens. As we have used the whole pathogen, there are chances the person may develop some of the symptoms, right, or it is comparatively less safe as compared to the second generation and the third generation vaccines, right. So, example of this is if we talk about the example, we are acquainted with the first generation vaccine, that is OPV. OPV is a oral polio vaccine. Oral polio vaccine, right students? So, this is a first generation vaccine. OPV, that is a oral polio vaccines. These are inactive forms of pathogen, right, and they are less safe as compared to the second and the third generation vaccine. So, one important part, they are less safe because there are chances that all the pathogens are not inactive and the person may develop certain disease. So, they are less safe as compared to the second and the third generation vaccine. So, this was the first generation vaccine. Now, we will be talking about the second generation vaccines. Second generation vaccines. In the lower classes, you must have studied about recombinant DNA technology and genetic engineering. Hope you all know about the recombinant DNA technology and genetic engineering. That's second generation vaccine. And this is number 2, that is the second generation vaccine. As the name indicates, second generation vaccines, and these are those vaccines which are created by recombinant DNA technology and with the help of genetic engineering. In this, what happened, we use certain medicines which are cultured in the laboratory, and for example of this human second generation vaccine is hepatitis B vaccine. Hepatitis B vaccine is created in the yeast cells, right. So, these are created with the help of recombinant DNA technology. In this, what happened? We have used one term, that is recombinant DNA technology. This recombinant DNA technology. In this, what we do, we take out those surface antigen of pathogen, right. We take out that which DNA fragment codes for that particular DNA fragment. And with the help of recombinant DNA technology, in this, when we take out those DNA fragments, we take out, while we are taking out that, we culture, we basically increase the surface, increase the whole the DNA with the use of PCR, that is a polymerase chain reaction, and by use of certain vectors and restriction enzyme, we create those vaccines that comes under the recombinant DNA technology, right. Other than this recombinant DNA technology, that is mainly we use genetic engineering, right students. So, what is the genetic engineering? We are manipulating the gene, right. We are manipulating the gene, or we are increasing in quantity, right. We are increasing in quantity and we use certain vectors, right. By using genetic engineering. Right students. So, example of this is hepatitis B vaccine and these are made in yeast cell, like these are made in yeast cell. Right? We used to make this in yeast, right? So this was the second generation vaccines. Next we have a third generation vaccines. Third generation vaccine, as the name indicates, third generation, right? So it is comparatively more advanced as compared to the previous one. For example, of this DNA, for example, are the DNA vaccines. And DNA vaccines are synthetic and they are potent as compared to the first and the second generation vaccine. And the third we have is third generation vaccines. The third generation vaccines, for example, are the DNA vaccines.